In this video, we will discuss about role of Granzyme in aged CD8 plus T cells for cognition functions. So, so far they have found out that aged CD8 plus T cells will cause this, will affect negatively this cognition function of the brain. So, and from literature they are able to see that this Granzyme is also plays a role towards this cognition function. So they wanted to investigate whether Granzyme in the Aged CD8 plus T cells is causing these cognition functions, or it has any kind of contributions towards these cognition functions or not. So for that, first they analyze this. First they checked the cells that are known to express Granzyme are present in the Aged CD8 plus T cells or not. So they are able to find out that this age-associated CD8 plus effector memory T cells are present, and this age-associated effector memory T cells is correlated with the high Granzyme expressions and can also be seen from their transcriptomic data. And these things, once they find out that those populations are present, now they wanted to compare whether there is a difference in the Granzyme expressions in this age CD8 plus T cells and the young CD8 plus T cells. So they checked this Granzyme expressions in two different populations, and they are able to see a significantly high expression of this Granzyme in the aged CD8 plus T cells compared to the young CD8 plus T cells. Afterwards, they checked these Granzyme expressions using flow cytometry also. From that, first they checked the activated cell signals that is known to express Granzyme, that is CD69 plus and CD8 negative. These cells are significantly higher in the aged CD8 plus T cells. And afterwards, they checked these Granzyme expressions in these activated cells, and they are able to see that these Granzyme expressions are significantly higher in the aged CD8 plus T cells compared to the young CD8 plus T cells. So from till now, they are able to confirm that the Granzyme expressions are higher in the aged CD8 plus T cells compared to the young CD8 plus T cells. They might have a role towards this decreased cognition functions in the aged CD8 plus T cells related to adaptive transfer. So in order to confirm that hypothesis, what it is, they did not directly use the aged CD8 plus T cells. However, what they did, they used the young CD8 plus T cells and overexpressed this Granzyme. And for the control, they used young CD8 plus T cells and express this GFP, and these things they did it before the adaptive transfer of the T cells into the mice. So once they have this cell system that has the young CD8 plus T cells plus GFP and the young CD8 plus T cells that overexpress Granzyme, they did the adaptive transfer after depleting CD8 that are present in the mice using CD8 antibodies, and then they did this analysis, RAWM analysis for the cognition function. And what they are able to first, they checked whether the Granzyme is overexpressed in the mice or not. From the panel 5, they are able to see that from the panel 5 the result shows that these Granzymes are overexpressed in the case where they used Granzyme overexpressed young CD8 plus T cells compared to the control. So once validating that their system is working as expected, now they wanted to see whether it has a role, it has any impact on this cognition function or not. And when they check this error rate in this RAWM analysis, they are able to see that error rate in the case where they used young CD8 plus T cells with overexpressed Granzyme are higher compared to the case where they used young CD8 plus T cells plus GFP that is overexpressed. From this results, it looks like this Granzyme has a role towards this impair or weaken this cognition function of the brain. That is evident when the Granzyme are overexpressed. The result shows high error rate in the RAWM test. So far, we have discussed these panels in the figure 6, and I hope you understood how they till now how they come up to that point that Granzyme might have a contribution towards weakening of the cognition function in the aged CD8 plus T cells. if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.